Huntington's community.
Stay away.
I couldn't stay away. So yeah, everything I say is my opinion, nothing to do with my company. So just a little disclaimer there.
All right, let's go. So I always like to start with the discovery of the Huntington gene, just because as a family member getting involved, I don't know if I mentioned that, but I'm also a HD family member. But when I first got into HD research, I learned about this amazing collaborative research field. And I always highlight this paper because it's quite a big deal in science to have a paper that doesn't have authors on it and just has the Huntington's disease collaborative research group. So I like to highlight that. And this was a really huge global effort. 58 scientists, over 12,000 family members give DNA samples that allowed us to, I say us, not me, but we now know that a single gene, the Huntington gene, which you've heard a lot by Rachel's amazing tour de force of kind of the basics of the HD 101, this Huntington gene leads to this Huntington protein. And we know the cause of Huntington's is really based on just too many letters of this CAG repeated, over 36, causing a mutant form of the Huntington protein. And we now know that the relationship with the number of CAG, so it tends to lead to earlier onset. And it also, we know that the Huntington protein, when it has that longer CAG, it's more sticky, it clumps, and it causes brain cells to die. And it generally disrupts a lot of the functions in our cells. And we now know that Huntington itself is a super important protein that is involved in so many different parts of our cellular machinery. We've known for a long time about the 50-50 inheritance of HD, unfortunately, for some of us. This is because Huntington's is one of those kind of diseases where basically we get 2 copies of every gene from our parents, 1 from each parent, and with Huntington's you only need 1 copy to have the disease. So we know a lot, but if we really look at this, this is a graph showing kind of age of onset with how many CAG someone has. If you focus in on the most common CAG repeats that people have in the HD community, which is around 40 to 45, you can see here quite a lot of variation, up to 40 years difference in someone with the same length of CAGs. So CAG isn't explaining everything, but there might be other factors that are inheritable that could be modifying or having an effect on how early or late someone develops the disease. So you got a little bit about this from Rachel, but just to go over things, because I think it's good to kind of refresh our heads. So our genome is basically the book that is you. It's the entire recipe or instructions that make us who we are. And every cell has the entire genome in them to give the instructions to make you. And then the genome is actually broken up into kind of chapters of a book, what we call chromosomes. And each gene is kind of like a paragraph within those chapters. But you can also think of the genome like a city like New York or Manhattan, where you have different boroughs. So the chromosomes are like boroughs, and the genes are individual buildings within, or individual blocks within those areas. And the way kind of DNA and genetic information is translated or propagated is through these letters and sequence. So we have the CAG that causes Huntington's, but the sequence matters in terms of what each protein and thing does. And they use these specific letters A, T, C, G, and but there's lots of variation in our genetic code. So sometimes there could be a single change that doesn't cause a disease, but is more common in certain populations than others. And we call these SNPs, so a single nucleotide polymorphism, so we just call it SNP for short to make it easier. And they can act like labels or tags that can be really useful to understand whether different areas of our genome are relevant to different diseases and that. So researchers can take all of these SNPs and search for them across the entire genome. And then they can see whether people who have an earlier onset or later onset have more of one of these SNPs. And that is called what we call a genome-wide association study, or GWAS. So I'll say GWAS from now. And to do these kind of studies, you need tons of DNA and tons of samples from people. And luckily, and since the gene's been discovered, we've had lots of research studies, or what we call the observational research studies, where people have given samples of DNA over many years, and we've been able to use that to do these kind of association studies. So one of the first initiatives was JOIN-HD, which combined a study in America called PREDICT-HD and a study in Europe called Registry. And they looked for SNPs that might be relevant to early or later onset. And if we think of our kind of the Manhattan metaphor, you can think of each of the buildings as a SNP, and the height of the building is telling us about how associated or how strong the association is with early or later onset. And actually, this is what the data looks like. So it's actually called a Manhattan plot for this reason. You get these peaks, and this is what the GWAS looks like in Huntington's disease. And what came out of this is different names of proteins, or the SNPs, where they were located was in genes that are, you're going to see these names more and more over the years. So MSH3, FAN1, MAHC. It's not very easy to keep up with because I get lost, and a lot of the science conferences talk a lot about them now, and I'm not an expert, but we have a few in the room. But what's interesting about these, a lot of the hits were from proteins that are involved in maintaining and repairing our DNA, so keeping it intact, which is really important. So I'm going to pass on to Emma, who's more of an expert on this. So
thank you. So I'm going to talk a little bit about how DNA repair affects the age at onset of Huntington's disease. And the short answer, it does this through expansion of the CAG repeat. So we sometimes refer to that as somatic expansion. So when I say expansion, I'm basically describing the CAG repeat lengthening process. And when I say somatic, I'm basically referring to any cell in your body that's not involved in passing on genetic information to the next generation. So what's important to note is that somatic expansion doesn't occur all over our bodies, but somatic expansion is detectable within gene carriers within their blood, brain, and in their liver as well. So as you can see, this person inherited, how do you use the?
At the top.
There we go. 40 CAG repeats, so they have in their brain at that point of birth, all of the cells in their brain have 40 CAG repeats. But as time goes on, the repeat basically lengthens over time, and then it eventually reaches this point where it becomes problematic for cells. And that cell will eventually be lost, and then essentially once you've lost enough cells, that's when motor onset will begin. So evidence actually existed for somatic expansion as a driver of disease long before the GEM-GWAS study. So this image is from a paper from 2003, so over 2 decades ago. And what's important to look at in this image is the 2 bands at the bottom. So this is wild-type Huntington, this is mutant Huntington. And then what's really interesting is these really much longer blobs, which basically represent these rare large expansion events. And you can see that they're only detectable in the striatum and the cortex, which are brain regions which are vulnerable in Huntington's disease, whereas in the cerebellum, there's nothing there. So then 6 years later, another group showed that more extreme expansions were shown in patients with an earlier disease onset. So this person has an inherited CAG length of 47, and they onset at 41 years old. And then this individual also has 47 CAG repeats, and they onset 16 years before the other person at the age of 25. So what's important to look at in these images is these pink bars, and you can see there's way more pink bars for this individual, meaning more somatic expansions. So because this idea had existed for quite some time, it actually meant by the time that the GEM-GWAS data came along, a lot of the mechanistic work had already been done. So there'd been decades of cell and animal research already highlighting the role of these DNA repair genes in the somatic expansion process. But before the GWAS had been done, there was a lot of debate over how important somatic expansion really was for disease. Was it just a byproduct of something happening, or was it actually driving the disease? So now that we have data from patients that shows that these DNA repair genes are important in determining age at onset, it really gives a lot of evidence for how somatic expansion is something that's important to target. So how does DNA repair drive somatic expansion? We're still sort of hashing out the nitty-gritty details of that, but we have a pretty good idea. So what we've landed on currently is that these repeat tracks form these unusual structures. These structures are then recognized by a DNA repair protein called MSH3. MSH3 then recruits other repair factors to the site, which are meant to repair it, but actually by accident end up making it a lot worse and basically causing the repeat to lengthen. So this is just again showing that over time it gets longer and longer and longer until it reaches a point at which it becomes toxic to cells. Cells will be then lost, and it's eventually, when you've lost enough cells, that's when symptoms will begin to show. So just to really simplify that down, you can think of MSH3 as a road hazard reporter. So MSH3 has detected this pothole in the road being the CAG repeat. So MSH3 calls in these additional repair workers, and what they're meant to do is fill in the hole, but instead of filling in the hole, they keep on pouring asphalt on the ground, and they're making this road longer and longer and longer. And the road is being built towards a nature reserve, and you can think of the nature reserve as the toxic threshold, or the point at which it becomes damaging to cells. So we don't want this road to reach here, because it could be damaging to the surrounding ecosystem, with that being the cell. So then we have this genetic modifier FAN1, who you can think of as a concerned citizen who's doing everything he can to stop this road from being built. So FAN1 is good, we want more FAN1, and we want less of these factors here. So what I was doing back at UCL, and with which some of my colleagues in this room are still doing to this day, is testing potential therapeutics using the most relevant cells that we can. So what we do is we take stem cells from a patient with Huntington's disease and we turn them into disease-relevant neurons. And then once they're neurons, we treat them with potential therapeutics and measure what it does to CAG repeat change over time. So this is just an image that's a really nice, pretty picture of what our cells look like before we give them the treatment. So just to recap, so genetic modifiers of Huntington's disease, what they don't do is cause Huntington's disease. What they do is determine when motor symptoms begin. They help to identify therapeutic targets other than Huntington, and they control, these specific DNA repair factors control the rate of somatic expansion of the CAG repeat. So back over to Lauren.
So I'm adding this quote from Robert Persifici, who's the Chief Scientific Officer of CHDI. He says, "There's nothing more precious to a drug hunter than an observation made in the population you want to treat." And that's kind of the big thing about the GWAS is they were kind of nature's clinical trial. These are people that seem to have these different variations in their DNA that have helped either modify their disease onset. So and this is already being translated, and companies are already starting to work on these different targets. We have Locus 23, which had an announcement last year where they've got big funding to take their target MSH3 to the clinic. So hopefully in the next year or two, we may have a program, first-in-human program of drugs that are targeting MSH3. And they are trying to do this using small molecules, so things that we can take as a pill, which is super exciting. And if you want to know more about that, myself and Ashley did a Breaking Down Barrier interview or casual conversation with David Reynolds from Locus, where we talk about that information in just a casual conversation. But there's a lot more to come. There are many companies working in this space, and this is just a few of them that are all have different programs that are developing targets that are relevant to DNA repair. And as we think about how we get this into the clinic and when's the right time to treat, a lot of the trials to date are in this manifest HD stage, but as this seems to be such a fundamental mechanism early on in disease, we really believe that targeting earlier might be important, and we hope that with the development of the HDYSS, which you've learned a bit about earlier, is the integrated staging system that allows us to look at stage people from the point of birth when they have carried the gene or not, and really start looking at these earlier stages way before someone's symptom onset. And hopefully, at some point, we'll be able to go into trials in people that are very far from onset and prevent HD from ever happening. But to do this, the issue with a lot of the clinical rating scales and things that are used as endpoints and outcome measures for these trials, they're not really useful in people that don't have symptoms because they're designed to measure things that you can see and a clinician can see in a patient. So we need more sensitive tools, and that's where biomarkers come in. They are something you can measure in the body. It could be blood test, or it could be an MRI scan or an image of the brain. They tend to be more sensitive to earlier changes. And I'll just highlight, I know Mina had a wonderful talk earlier. If anybody went to it, you can probably catch it. If not, highlighting some of the results from the HD Young Adult study, which is a very unique cohort who were followed for over 5 years, and they were at baseline over around 24 years from their predicted onset. But they also saw some hints to somatic instability being a potential driver that early in disease. So the team are here. I see some of them in the audience. Please go to their booth and talk to them, Mina, Kate, and Michaela, and their new clinical fellow, Lisa. I'm sure they'll tell you more about it. So just to finish, this is some images from my own research, and the people that take part in our studies. Me and my lab are also trying to look at biomarkers early on and use ways that we might be able to do home testing for NFL or potentially develop that for somatic instability as well. But I just want to finish and highlight all of the participation in these to advance these things has been a huge collaboration between scientists and researchers. And I think we're still the Huntington's disease collaborative research today. And I hope that you come away from this weekend and feel really empowered to take part in whatever studies that you can, because it's all getting us a bit further down the line to hopefully one day having prevention studies and a drug that prevents HD. And thank you. So we'll have time, I think, for some questions as well, if anybody wants to. We also have another expert in the room who can talk with me a bit more about PMS1. Yeah, Hayley?
So with your, I could just shout, I won't. So just with your pothole analogy, so just, this isn't kind of just about HD, but just in general. So when the pothole occurs or the damage occurs, like, that's just normal, that happens to everyone in all different genes?
I think there's something specific about repeats.
Yeah, so basically someone with a gene carrier has already an expanded CAG repeat tract, and there's something about that 35 repeat threshold that we think causes that CAG repeat tract to form these unusual structures. So you wouldn't get those structures in genes that don't have the expansion. Yeah.
And you also have similar kind of phenomenons in other expansion. So there's other diseases that have different proteins that have CAG expansions or other kind of letters that are expanded that seem, it might be a mechanism that happens in all of them.
And those DNA repair modifiers, some of them are modifiers of those other diseases as well through somatic expansion.
Okay, this is just a question about kind of like the brain neurons and synapses. So, like this week I learned that HD is also, it's not just like, I guess the glial and other cells like dying. It's also because of like excessive synapses connections being cut? Is that? Oh, okay. Because I know that like synapses with neurons, like as you grow, your brain will automatically cut the ones that you don't need anymore. So I was just wondering, with HD, is there a connection between like the mutant like overcutting the synapses, which would cause?
So there's definitely evidence for less synapses and less connections in the brain, but whether that's because Huntington's like pruning those more or the fact that those cells are dying, so there's less connections, I think it's still, but I am not an expert on that, so I can try and find someone. I don't know, did you have something to add to that? Yeah, but there's definitely evidence that synapses get lost in HD. How much that is to do because of Huntington itself impacting those, or the fact that the cells are dying anyway, it's hard to say. Yeah.
Hello. I was just wondering, kind of with thinking about Huntington lowering and also targeting somatic expansion, both as therapeutic targets, like how do you think that they might play together? And is it possible to have, like, to take a gene therapy for Huntington lowering as well as something for somatic expansion?
Yeah, if you want to go for it.
I think there was some recent data that came out at CHDI this year that showed that Huntington lowering alone and MSH3 lowering alone so MSH3 lowering being somatic expansion targeting, had like beneficial effects on Huntington's disease. And then when they did it together, they had like synergistic effects. So combining it was even better than either one alone. I don't know if you want to add anything.
Yeah, so I didn't mention it because of time, but one of the other hits on the GWAS is actually on the Huntington gene itself. There's a thing, so it's a bit more complicated, like everything in science, that we have the CAG repeat, but at the like penultimate, before the last repeat of CAG, there's a CAA. So when the machinery makes that instructions into a protein, it doesn't make a difference to the protein because they code for the same kind of building block. But for some reason, we think it's linked to somatic instability, that when you lose that CAA, it makes that repeat even more unstable. So that's one evidence of, oh, Rachel's here as well. You can answer some of these questions. Oh, really? Okay. Yeah. I was not paying a lot of attention at the CHDI, so let's be honest. But also, there's evidence that Huntington itself goes to places where there's DNA repair happening or DNA damage. And there's built-in evidence. I know you find stuff that there might be a DNA binding part of the Huntington protein. So there's something happening with DNA itself in that process that's still to be understood. But yeah, I would listen to Rachel. She's very smart and knows more about this.
So in the clinic, I guess that when we see people who are thinking about testing, they're asking us when they might develop symptoms, what the age of onset might be for them, and that sort of information would be really helpful. And we have absolutely no way of predicting somebody's age of onset based on the information that we currently have. Do you think that eventually this information might be helpful to us?
Absolutely, but as you know, like we don't have enough information to give an individual person. So all the stuff that we find in research is big groups of people where we're seeing associations, and a statistical association in a big group of people isn't like going to you and saying, you have this modifier and means you're going to have this many years. I think maybe what will develop is maybe risk scores and things like that, but how meaningful they will be to an individual. I think what we didn't mention is like different modifiers have different levels of impact. So some of them have a stronger effect on the changing onset or not than others. But yeah, more data. That's why, as something like Enroll grows, that's like 20,000 DNA samples. It means everything we understand about these modifiers will continue to grow and develop over time. But I think someday, yeah,
I think also we've got to remember that, like, when you control for CAG repeat, 40%, I think, of the remaining variability is heritable, but that means that 60% isn't heritable, so we can't predict it by genetics. And there's lots of lifestyle factors that we can't account for in those prediction scores. So I think it's an important thing to be able to do, but it's going to be really tricky.
Maybe this person first.
Yeah, just someone else.
Okay, hopefully this question is relevant. correct me if I'm wrong, but I think in diseases like sickle cell anemia, it kind of was maintained in the population because it had protective effects for other illnesses. Like malaria, that's what I was thinking of. Have you found anything like that with HD, or are there genes that have been discovered that are protective against HD, if that makes sense?
So there's evidence, and a lot of people have the thought that there's an evolutionary benefit of having longer CAGs. So if you look at how organisms have developed over time, the simple organisms, I didn't just say that, did I? Oh dear. Organisms, which were simpler, that didn't have a nervous system, didn't have Huntington's. Having Huntington as a protein kind of coincides to when organisms develop nervous systems. And then more complex nervous systems like mammals coincides with them having more CAG repeats. And then, there's lots of different people saying different things. Like someone's put CAGs in a plant and it's made it grow 3 times the size. And so whether it protects from other things, but there just seems to be a propensity for these to repeat, and there might be some benefit, but I don't think there's a concrete evidence either way. Probably time for one more, or we're good.
Yeah, I did, but if someone else has one, it's fine. Yeah, it was just the question with the 3 brains, and you had the numbers of the different CAG repeats, and obviously it increased. Has that been, how's that tested? Because I know we were talking in the HDYO talk about obviously brain biopsies and things to obviously know where the different repeats are, and I think you had something where one was 373 or something. So if you could just briefly touch on how you kind of measured that and know that.
So that's from post-mortem data from patients who have passed away at different stages of the disease. Did you want to add anything?
So there's some exciting new evidence where different groups have done single cell methods as well. So there's new advanced technologies that allow people to take every single cell and barcode it so you know exactly what type it is, where it comes from in the brain, and they were able to check about the other things that are expressed there, as well as the length of the CAG, so they can see how length of CAG over time within a person in different cell types and how that impacts other things. So it's
also from post-mortem, right?
Also from post-mortem, yeah. So caveats all around.
Cool, thank you.
Yeah.
I was wondering kind of what's the journey between identifying a gene modifier and a pharma industry targeting that gene modifier. How does that look? Patients are usually your family members, very interested in knowing how bench work translates into pharma companies. I don't know if I'm explaining myself well.
No, I think I understood. So you're asking about kind of how does a finding like that from a GWAS translate to finding a new drug that targets that, and then so there's a couple of things. So when you have new evidence, that then got a lot of groups interested in looking at this. So they look at the genes that came up in those hits and try and see what they do, different kind of experiments to see what about that gene might be interacting with this. So they might like knock out part of the gene and do this. So it helps them find out what actual part of the protein is involved and what. So that's what the kind of work that Emma and Joe would do in cells. Some people do it in mice, and they try and see how is that actually working. Then companies might have an idea of whether they need to up or down, like push it up. So Emma's analogy, MSH3 and things they want to reduce, and found one. The other issue with a lot of DNA repair proteins, DNA repair is really important, and you don't want to. There's certain ones that you don't want to play with because when you impact your DNA repair, you can the wrong way. You could cause things like cancer or things. So it's very important to pick your targets. So there's evidence from other fields like cancer about these DNA repair proteins. So researchers had a good idea of like what would have been more appropriate targets than others and what would be safer. Yeah. So there's a lot of it. So that's why MSH3 comes out more, not FAN1 as much. That's how a lot of those decisions have kind of come around. Does that answer? Yeah. I think. Yeah, amazing. We're all done. Feel free to talk to us after.